All right, guys, so we are getting ready to talk about our next electrophoresis lab, and what you're seeing me do right now is flip through the very large lab document, by the way. You have watched the pre-lab video at this point, probably, and you kind of have an understanding of what this lab is all about. So what has happened here is that this is a forensic chemistry course. So what that means is that in our previous electrophoresis experiment, we focused on identifying parents from children who maybe have been separated from their parents in some form or fashion, whether that's through migration problems or whether that's through war times, or that scenario, of course, was written for a wartime. But children get separated from their parents quite often, and this was one of the ways that we could help determine who's this who's the mother, who's the father, is there an overlap, is there something there that kind of commonly goes across the board. So in this particular electrophoresis experiment, we take a different turn, and this one is going to be focused more on the crime scene. So here is a scenario where we went out to a crime scene, or someone else did that, and they obtained DNA from a crime scene, and that DNA could come from many different things. It could come from blood stains, it could come from skin cells, it could come from hair. All of those are going to be listed here in the background of the document, and that's what we're seeing here. So these samples have been brought into the laboratory. We are not the ones that go out and get those samples. We are the ones that stay in the lab in a safe, clean environment, and we are processing these samples as they come in. So a crime scene sample has came in, and then someone, maybe us, we have taken suspects, different suspects. There were actually a total of 3, I believe, if I'm not mistaken, not 2, but some suspects' DNA, and we've kind of isolated those as well, and we're going to be running those on electrophoresis. So what happens here is that the DNA samples that are collected from the suspects or from the crime scene, it's probably not going to be enough DNA for us to reliably do something with it. So what we have to do is then amplify it or multiply it. And when we multiply the DNA, we are going to multiply this by many fold. I mean, we're talking about like multiples of million, if not a trillion fold, in order to get something to run on the electrophoresis slab. And in addition to that, I also want to say that this process, amplifying it, multiplying it by who knows how much, is going to come from the process of PCR. So polymerase chain reaction. So we have taken these samples in the lab. We have now amplified them, PCR'd them, and this was done off camera. You did not see this. We obtained the samples after the PCR took place. That's kind of where we started and picked up. But in the PCR, this is where the DNA was split. So those 2 double bands were ripped apart. And then we added 3 different temperatures, and we added tack, and we added primers, and we added bases, and we allowed the replication to take place over a course of time. So heat up, cool down, heat up, cool down, heat up, cool down, heat up, cool down. And then these were the samples that we have taken into our laboratory that we have ran. So up until this point, we have taken all of that for granted. So those samples that we injected in our lab video, those were already taken from the crime scene or from the suspects. Those were already PCR'd by the time that we got a hold of them. And it was just really up to us to inject those samples into a gel and then begin to analyze them and undergo electrophoresis with them. So that's what we did here. We went through the electrophoresis. We went through the recipes. We hooked them up to the voltage chambers. We analyzed them, we stained them, and then we destained them. And we ended up with a gel, and that gel needed to be stained because we could not see the base pairs, and that's normally kind of what goes on. And unlike the previous one with Who's My Father, these were not just simply dyes just to get you into the habit of what electrophoresis is all about. These were actually DNA bands. That's what these were. So it's going to be more true to what we would see in a real environment. Here were the samples that we injected. So number 1 is the ladder. number 2's the crime scene sample, and then 3, 4, and 5 were suspects 1, 2, and 3. So we need to figure out if either one of these suspects were at the crime scene. That does not mean that they did the crime. It just simply means that they were present at the scene. So there's no way that we can prove if this person pulled the trigger. There's no way that we can prove maybe if this person, you know, chop, 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 and hacked. But we can at least say, well, they were at the crime scene because their DNA showed up. Their DNA maybe was on the gun, maybe their DNA was on the knife, but we can't really necessarily say that they pulled the trigger or they stabbed somebody because we were not there and we did not see that. But we can at least put them at the crime scene, and maybe that's one step in the right direction in order to find someone guilty if they really did do it. So in the previous video, we went through taking the gel out of the chamber. Then we went through staining the gel and then destaining the gel and using our orbital shaker. And then there toward the end, I kind of showed you what the destaining process looked like, where the water was somewhat clear. It had a little tinge, very, very slight blue, but it had a little tinge of color there, but not much at all. And then what we ended up with was a gel that looked like this one. Now, I realized after the fact that the picture doesn't quite do justice. We don't have a proper imaging system in-house. So I just kind of have to deal with the camera. And if the camera can pick them up, then great. Sometimes it can do that and sometimes it can't. And that's what you're seeing over here on the right hand side. I have drawn a extra little band that is here because that actually showed up in-house, but it does not show up quite well on the picture that I have taken. The other thing I want to talk about is, I almost let it run too long. And the reason is because I've got some banding that's happening down here at the bottom of the gel. That's where the DNA ladder color was and the sample loading dye color was. So with that said, I kind of ran it until it got closer to the bottom, but I should have stopped it a little bit earlier. That way it would have prevented some of that from happening. But overall, I can see bands. They're separated. They look pretty decent. And we need to go through and we need to actually do some measurements now. So the very first one is the DNA ladder, and the ladder is made up of the base pairs. That's why we run these ladders. They're all base pair driven, and it's a assortment of different base pairs. And I will give you those base pairs in your laboratory worksheet. You will see numbers that are there with the base pairs, and those numbers have been provided, and you will need to do something with those numbers. So in order to do something with those numbers, we need the distance of how far they've traveled. So what this means is that up here at the very top, this is where I've injected a sample, and the sample has migrated down the gel closer to that red cable, and then this particular piece stopped right there. So this gives me an idea that that piece, whatever that piece is, it's probably larger than the rest of them. And the larger ones move slower. So that's how I know that compared to all of the other bands that show up on the gel, this one is the largest because that one moved the slowest. That one was more toward the top of the gel itself, okay? And then there's other bands that are there. So they begin to separate out, different chunks separate out from each other. And then we actually even see bands that are like down here at the very bottom, right? There's a band that's right there. And that is probably a smaller band, and that smaller band is present, and that smaller band is going to travel the furthest, and here it almost ran off of the edge of the gel. So we need distances. That's what we need. So here is the distance for spot number 1. And if we go over, you can see my ruler, my centimeter ruler. And the only thing you got to do is just record the number of centimeters. I'm going to let you make that decision. Record the number of centimeters that you see band number 1. Then we see band number 2 and band number 3, band number 4. Band number 5 is actually right here. It's very faint, but it's there. Band number 6 is right here. Again, very faint, but it's there. and band number 7 is actually down here at the very bottom. So once more, my camera did not show that, but in person I saw that a little bit better. Does destaining help with this process? Yeah, it does sometimes, but actually just having a proper camera with a very bright white light background will make these show up even more so, especially with the camera and a document station that we do not have currently in ChemTech, but hopefully maybe sooner or later down the road we'll get one. All right, so there is our lane number 1, and lane number 1 is our DNA ladder, and it looks like there are 7 bands that show up in the ladder. Next one over is the crime scene. So I need to do the same thing here. I see 2 bands in the crime scene, right? I see one that's going to be present there that shows up pretty well. And I also see a band that shows up here as well. So there are the base pair fragments, so different sizes, that showed up at the crime scene. So that person's DNA cleaved in 2 different sizes, and those 2 different sizes separated on the gel, and we can measure how far down they traveled, and that distance is going to be very important. Next up is lane number 3. And with lane number 3, I kind of see suspect number 1. And if I look at this gel, I definitely see 1 band, right? I mean, there is 1 band that is here, absolutely. And that's really all that I saw as well in person. Nothing really else to talk about with lane number 3. And suspect number 1's DNA was cleaved, and it was cleaved in 1 base pair fragment, and that's basically what showed up. So once more, I'll let you make the measurement distance, and you can record that centimeter, and then we'll move on to the next one. All right, in the next lane, we've got suspect number 2, and suspect number 2 just as well. We obtained their DNA, we amplified it, we broke it up into fragments, and this person fragmented in 2 different locations. So it looks like this person fragmented here, and you can make that measurement, and that person fragmented there as well. So you can make that distance and judgment call just the same. So once more, like all the rest, you need those centimeters because those centimeters will help you identify the base pairs of these fragments that are showing up on the gel. Finally, in our last lane, this is suspect number 3. And in suspect number 3, I did have a little mystery peak that happened down here at the bottom that did not show up very well at all. But just as always, here is the well. That's where we started the sample, and then the sample migrated down. So this is where one of the base fragments stopped, and right here is where the other base fragment stopped. So I will let you record those distances as well, because once more, just like the rest, you need those distances in order to do some of the laboratory work. If I go back and I take a larger look at the overall view of the gel once more, this is the gel from the crime scene lab experiment. So just as we've discussed, the first one is a ladder. The second one is a crime scene. The third one is from suspect 1. The fourth one is from suspect 2. And the fifth one is from suspect 3. So the goal of this lab experiment is to determine if either of these suspects were present at the crime scene. So unlike a paternity test, if we want to know if they were present, then that means that their banding should show up, and it should be a perfect match, right? The parental DNA lab exercise made you match half and half. So half comes from the mother, half comes from the father. In crime scene identification, we are trying to determine if that person was present at the scene of the crime, yes or no, which means that that person's DNA would be present at the scene of the crime, yes or no. So we are looking for perfect matches in these types of scenarios. And I think visually, by looking at the gel, you can quickly determine if those scenarios take place or not. But that's not the only thing that this lab experiment is going to get you to do. So what else will happen? Well, just as in the lab video, the pre-lab video, you will go through the motion of making a calibration curve. And the calibration curve will be based on the number of base pairs as well as the centimeter or the distance that those base pairs traveled. So you've got to figure out, well, which one of these is the x-axis and which one of these is the y-axis. And do we have to modify anything, right? That's the other thing. Do we just plot straight up base pairs? Hint, hint, hint. Do we just plot straight up centimeters? Well, we need to do Excel, and we need to do a regression line, and we need to go in and do a y equals mx plus b. That's what we're trying to do here. That way, when we use these distances, we can talk about a y equals mx plus b regression line, and we are able to put in the distances that these fragments traveled, and we can plug those into the linear regression equation the proper way. We can then solve for the base pairs, the number of actual base pairs that's located in that band. So we can go one step further than what we did in our previous experiment together. Actually, a couple of steps further. We're looking at real bands that traditionally show up in electrophoresis. We are measuring distances because we didn't do that in the other one. We just compared colors. We're plotting a calibration curve in a way based on the base pairs that we are using. So that's something different. And now we can use the distances that have been traveled for each one of these bands. The distance between here all the way down. The distance between the starting point all the way down. And we can plug that distance into the regression line and solve for the actual number of base pairs that show up in that fragment. Right? That's something that we did not do in the previous experiment. And that is how this is going to push you a little step further in the proper direction of gel electrophoresis. Okay? All right, so that is the summary of the DNA crime scene. Oh, excuse me. That we have done a PCR on. So, as a summary, lane 1 was the ladder, lane 2 was the crime scene, lane 3 was suspect 1, lane 4 was suspect 2, and lane 5 was suspect 3. A simple case of who done it. That's what we're after. So did either one of these suspects do it? Well, we can't really make that determination, but we can maybe put them as present on the scene of the crime or absent at the scene of the crime. Maybe all 3 are guilty. Maybe none. Maybe one of them. Maybe 2 of them. Your electrophoresis will help you determine that. All right, so with that said, good luck on plotting those graphs. Good luck on generating that data. Go back, review the pre-lab exercises, review the lecture material if you need to know how to do this. That's where you're going to find all of those little hidden gems of nuggets of information on how to complete this lab the proper way.